Hey everybody. Unfortunately, because of the snow day, we weren't able to do this in person, but I'd like to set this up. If you follow along, you should be able to take your sequencing data and figure out at least what your species is and maybe some of the uh, evolutionary relationships it has. All right, so you're going to follow the link in the protocol, and you're going to end up here at DNA Subway. DNA Subway is a way to process the data and compare it to um, a large database of species that is already out there. All right. So we open up DNA Subway. You are going to create a project. Go back. All right, so it'll look like this. We're going to go ahead and create a project. You can call it whatever you want. All right. We want to have Sanger sequencing clicked on, and we go ahead and we create the project. Now, there's no files in here yet. So what you're going to do is upload from device. So what you should have already done is uploaded that folder that I posted for you on Brightspace, and you should be able to grab your data from there. So if we hit upload from device, all right, if you downloaded that data, it should be sitting there in downloads. All right, I've got a few here. So what I'm going to do is I'm going to select, we're going to do 133 is our sample number, right? You're going to download your sample number. And so what they have is they are going to have an R and an F here. And so there's going to be two files with your sample number. You need to download both of them. What you're going to do is click on the first one, go ahead and hold down the shift key and click on the second one. So now we've selected both of them. It's important that you select both if they are present. All right, so they are there. They should be uploaded. There are two samples. All right, so we're going to go to the next stop on the DNA Subway. All right, what we should see now is that, I went way too far ahead. What we should see now is trim the sequence. All right, we're going to work through these. You can go ahead and what we're going to do is you want to select a sequence to trim. Or actually, we don't need to select the sequence to trim. So that would be if you want to do one individually. We're just going to say auto trim all of them. Now what that's doing, so we talked about Sanger sequencing in the other video. What that's doing is there is on either end of the sequence going to be a lot of stuff that it doesn't resolve very well, and a lot of sort of junk there. So it's going to go in, it can recognize that and trim those off. Right? So all trims are complete. Now we're going to pair our sequences. So we can see that this one is an F and this one is an R. That means forward and reverse. So we're just going to click on the F for the one that says F and an R for the one that says R. We are going to go ahead and call this species 1. All right, that'll be important later on. All right, and so they are now paired as species 1. Now what we can do is go ahead and we can do what's called edit the consensus. So we select our species. All right. And so what it does is it's going to go through and automatically line up the sequence. Right. So it's going to line up your forward and reverse, right, those complementary bases wherever it can, and come up with the best fit as far as where your forward and reverse sequences line up with each other. So remember, when we make copies from way back, when we make copies of DNA using PCR, you've got two different primers, one for the forward strand, right, those anti-parallel strands, one for the reverse. And so we're taking and we're lining those two up with each other to see where our sequence is. And then we can go ahead and select our sequence, all right, our species 1, to go ahead and we click on BLAST. Now what BLAST is going to do, it's going to go into the NCBI database and search for any sequences that match our sequence. All right, so there's thousands and thousands in there. If our species is in there, it will go ahead and go in and find it. So once it comes up, you can go ahead and click on it. And we got a bunch of scientific names. All right, so each of them has different levels of match. All right. And so what you're going to do is you're going to take a look at the top 4, all right, matches that you got, and take a look and try to figure out what the most likely one is. All right, so the E value here is going to tell you how likely it is that this match is just by random. All right, we've also got alignment length. So this is the number of bases that lined up and the number of mismatches that there were within that. Right, so you can see the lowest number of mismatches and the longest alignment length is this Odontomia species. All right, if we click on it, it will show you, all right, a little blurb of what this actually is. All right, and you've got a Wikipedia link here also. So this is Odontomia SP, which just means random Odontomia species. All right, lines up COI gene most closely with that one. Right. So in your lab, what you're going to do is you're going to list the first 4 ones that line up. All right, and so this is going to tell us what our species is. So the other thing you want to do is go ahead and so for example, we've got Odomontosinca. What you're going to do is go ahead and Google your species that you find and determine, okay, does it make sense geographically? Does it make sense? Is its range include North America, that sort of thing? And so the idea being that if you've got multiple species that are showing up, which one's the most likely to be the correct one as far as, where we collected our species, has it ever been seen there, that kind of thing. Then what we're going to do is we are going to select the top 10 hits or so. All right, we're going to go ahead and add them to projects. All right, so now they've been uploaded. So what you're going to do is you're going to go in and you're going to add all the files that are there for your class. All right, so any of the ones that are already in there, go ahead and upload those. So you should have downloaded your entire class's worth, and go ahead and add all of those in there. So you should have a bunch of pairs in there, and we will go from there. Okay, so you can see an uploaded. You should have, all right, and now a whole bunch of other organisms in here. All right, so I've got a number that I put in from before, but you're going to see all of your classes in there. There we go, as forward and reverse. All right, so we're going to go to the next stop. So we're going to go ahead and say auto trim the untrimmed. Okay, so once you have them uploaded, just like before, we're going to go ahead and continue down the subway. We're going to auto trim. All right, it's going to go ahead and trim any ones that we didn't have in there. And so now what we're going to do is we're going to pair them again. Remember, select the R for the ones with R, F with the ones with F. And we are going to go ahead and give this a label. In this case, species 2. If we were in lab and we knew what each of them were, we could label these now with what the actual species were. So if we were doing this together, we could. But we can just put some names in there to go ahead and keep track of them. All right, so we've got our 121 forward and reverse. All right, I've got a bunch of stuff in here that you won't see because I've been uploading things previous to this. Oh, I forgot to name it. If you forget to name it, you can go in here, undo the pair. Let's try that again. Call this species 3. There we go. All right. So then we can go to, all right, we can show consensus. And hit next stop. All right, now we're going to go to BLAST. All right, so what you can do is you can select all of your new species, all right, so the entire class worth, and go ahead. So the other ones have already been blasted on mine, and go ahead and it will blast. Now, if this is taking forever, if it just keeps going, one of the things you can do that we found that helps is if it kind of seems to get stuck, you can just hit the refresh button and it'll go through. So we're going to go ahead, since it's taking a minute, go ahead and give that a try. All right. Back to blast. Oh, it's still spinning. So sometimes you just got to wait. And boom, there they are. Okay, so we've got our species in here now. And so what we can do is move along, and we are going to go ahead and under this analyze. So once we have our blast, we are going to click on muscle down here, so that if that doesn't open up, you should have that. All right. And so what we're going to do is we are going to select all the ones that we want to add in there. All right. So our species 3, species 2, add those in. Let's see. Pick one, let's see, of your species. And then we'll throw a couple other ones that are in here. All right. Then you're going to go ahead and click muscle. Now, this one might take a while. All right. And what will happen is this thing will pop up. And so this is each of the species. All right. And here is basically what it is each colored line represents a section that is unique to that species. The gray areas are the ones that align with other species. And so what you see is a unique pattern here of unique sequences for each of our species, right? And so what that tells us is how closely related these are, right? The more sequences they have in common, the more closely related each of our species is. All right, so you are going to go down to Philip ML here, and we're going to go ahead and hit submit. What that's going to give us is it is going to show you your phylogenetic tree of all the species that we added in there, right? Basic idea is the closer together they are on this, the more closely related they are. So like our fork-tailed damsel and species 2 are closely related. All right, our damselfly and our eastern pondhawk, they're both Odonata. They're both in the dragonfly family, so they are more closely related. Gives you an idea of how each of our species relates to each other in this group. All right, so I hope you all had some fun with this one. I hope you learned a couple things. Extremely powerful tool for examining the world, finding species, monitoring species, lots of different things that can be done with these databases and with our DNA barcoding. All right, we'll see you later. Good luck.